So now we're going to talk about the development of the bones. And there are two primary methods by which bones develop in an embryo. There's endochondrial ossification and then intramembranous ossification. For endochondrial ossification, the cells are first going to lay down a layer of cartilage which forms a template for forming the bone. In the case of intramembranous ossification, the bone actually sort of develops directly from the mesenchyme without first laying down a cartilage skeleton. Now, endochondral ossification is what we use for axial skeleton bones. Remember, the axial skeleton is your spine, your sternum, or your breastbone, and your ribs. And really, almost all of the appendicular skeleton, except for a portion of the clavicle, also develops via endochondral ossification. Interestingly, the pharyngeal arch cartilage is laid down in the same way, and if you remember, the pharyngeal arches are critical to the formation of the face and the neck. We will come back to them in the last week of class. There are certainly several different cell types that participate in endochondrial ossification. Chondrocytes, which are cartilage cells, osteoblasts, and osteoclasts will also be present. For intramembranous ossification, again, where the bone is developing directly from the mesenchyme without going through a cartilage stage, that's the kind of bone formation we use for facial bones, for the cranial vault, so some of the structures of the skull, and then again a part of the clavicle. In this case, the bone is derived from the neural crest cells in the paraxial mesoderm. In the case of muscle, there are a couple different cell types that contribute to the development of muscle. Initially, these muscle cells are called myoblasts, and those are the embryonic muscle cells, and they do migrate, many of them, around the body to develop muscles in various locations. You saw the diagram. There are lots and lots of different muscles. And they then differentiate into myocytes, which express actin and myosin, which are those contractile fibers. They're bundled, of course, as we talked about in skeletal muscle, into myofibers and then into even more complex bundles of fibers to allow for muscles to contract. Now, striated muscles develop in waves. First, you lay down a layer of embryonic muscle. That's the first wave. Those are the myoblasts, also called primary myogenesis. And then there's a second wave during the fetal period, which is where most of the muscle is laid down. And then, in fact, a third wave after delivery, where certain cells are triggered to become myocytes. But the primary time for laying down muscle, most muscles are laid down during wave 2, which is during the fetal period. So back to somites, because both muscles and bones are derived from somites. And somites are actually really interesting structures. They do a lot of neat things. But by way of reminder, the somites are derived from the paraxial mesoderm. And the paraxial mesoderm differentiates into somites. And so we saw the structure, they sort of follow the pattern of the future vertebrae along the spine and tail up to a point of the embryo. Now, the location of the cells within the somite determines what structures they are going to become. So the ventral medial cells will become the sclerotome, while the others will become the dermomyotome. So it's based on where they are in the somite. Now, the sclerotome and the dermomyotome. The sclerotome is going to surround the notochord of the fetus, and ultimately they will go on to form the vertebrae, and we'll talk about how that happens. But the vertebrae and the ribs are derived from the sclerotome, whereas the dermomyotome produces the dermis and then the myotome, which goes on to form the epimere and the hypomere. The epimere will go on to form the muscles of the back, whereas the hypomere will form the muscles of the body wall, the thorax, abdomen, and limbs. But we're going to go back to the sclerotome and highlight that particular process. Now, the sclerotome, so again, this is a subdivision of the somites. It has its own cranial and caudal ends, and that would make sense because we're following along the spine of the embryo. So there's a cranial end and a caudal end to every structure that follows that pattern. In this case, which end is cranial and which end is caudal is determined by the density of the cells in the region. That's how the sclerotome determines what it is, but of course it also follows the basic body patterns. So the sclerotomes then are going to split into their cranial and caudal segments, and the caudal end of one sclerotome will ultimately fuse with the cranial end of the next one, and those two together will form a vertebra. So in fact, the vertebrae are formed from cells from two different sclerotomes. And what's interesting about that is that reorganization, or what your book is calling resegmentation of the sclerotomes, alters the relationship of the units, in this case the future vertebrae, with the surrounding support structures, so that now the arteries and the nerves of the spine run in between the vertebrae instead of through the middle of the sclerotomes as they had been before. Now, interestingly, the most cranial four somites will fuse to form the occipital bone, which is the base of the skull. So while most of the somites go on to form vertebrae, a few of them do not. In the case of the ribs grow as extensions of the vertebrae, again via endochondral ossification. But basically the vertebrae put out two projections, one on the left, one on the right, that then sort of grow forward and meet in the middle. And when they get to the middle, they'll run into the sternal bars. So the sternal bars are mesenchyme that's sort of condensed into these two bars right in the middle of the chest. Ultimately, they grow together and fuse to become the sternum or breastbone. Although the ossification of the sternum, so the conversion of the sternum completely into bone from cartilage, doesn't complete until after birth. The first 7 ribs in your body will connect to your breastbone, assuming normal development, and the others do not. Development of the long bone, again, the development of the various bone types is a little bit different. We'll focus on long bones. But in the case of the long bones, the mesenchyme is going to condense and differentiate into chondrocytes, which are then going to secrete cartilage. So we talked about this process of endochondral ossification. We've mentioned it several times. So this is what's happening. We're laying down this cartilage skeleton. Now, we talked about the structure of long bones. In the developing long bone, the chondrocytes and the epiphyses are resting. Okay? But in the diaphysis, in the very center of the diaphysis, you have hypertrophic or fully differentiated chondrocytes. So they're not going to change into any further cell types. Their differentiation is done. And they are laying down cartilage. And that very middle region of the diaphysis of the long bone is called the primary ossification center. That is where you get a nutrient artery that has actually reached into the bone, to the interior of the bone, and is supplying nutrients to that region. That artery brings in osteoblasts and osteoclasts, which can then begin the process of bone formation. And then from that point, ossification proceeds peripherally in both directions, so towards both epiphyses. At birth, the diaphysis will be fully ossified, but as we talked about, the epiphyses remain cartilaginous up to a point to allow for new growth and lengthening of the bones. Okay. Now this is sort of quick and dirty for limb muscle development. We'll cover limbs in Chapter 20 in more glorious detail, but for the moment, just sort of a quick idea of how the muscles of the limbs come to be where they are. We talked about myogenic cells migrating, and in the case of the limb buds, that's how they get there. So 2 masses of myoblasts will form from migrating myogenic cells in each of the limb buds, and we'll talk about limb buds at another time. For the moment, just sort of assume they're there. But in this case, the myoblasts are going to organize themselves into a dorsal muscle mass and a ventral muscle mass, which will ultimately become the dorsal and ventral muscles of the limbs. And again, more on this in weeks to come.